Hello, are there any questions? If there's no questions, we'll begin shortly. Oh, let me get my microphone on, sorry. Let's see. All right, hopefully that is better. Okay, where's my syllabus? And let me see if I'm showing the screen. I don't think I am. Nope. Okay, is that better? Everybody see the screen? Can somebody please comment so I can get a confirmation? All right. Today we're finishing chapter 5. Hopefully we will finish. If not, we'll have to work on it next week. There is no lab today, but I will be showing up at 6:30 to 6:45 to answer questions. And if there's no questions at 6:45, I will be logging off. So if you have questions and you want to ask them, make sure you show up between 6:30 and 6:45. Of course, if there are questions, I will continue until the last question is answered. All right. Hopefully you took the quiz last Wednesday. There is no quiz next week. So we were talking about aerobic respiration. And I believe I had talked about this slide showing you the overview of aerobic respiration. Is that correct?
I think that's correct, yeah.
Okay. So aerobic respiration has 3 parts: glycolysis, the preparatory step, and the Krebs cycle shown here, and then the electron transport chain, which is really kind of shown here. And let's talk a little bit more about glycolysis. Glycolysis can also be called the Embden-Meyerhof pathway, after the 2 scientists who discovered it. I and most people don't call it that. We call it the glycolysis pathway. Glycolysis is the oxidation of glucose to pyruvic acid. And if you remember, aerobic respiration normally uses glucose. It can use other food molecules, but most of the time, at least for couch potatoes, like myself, the other food molecules will be converted into glucose, and then glucose will be sent down aerobic respiration, starting with glycolysis. Glycolysis is the oxidation of glucose to pyruvic acid. Glycolysis occurs in most living cells, and it can occur in the presence or in the absence of oxygen, because glycolysis does not use oxygen. It does produce a little ATP, and a little NADH. And I'll talk more about NADH in a slide or two. This is showing you the general steps of respiration and fermentation, and we'll talk more about fermentation later today. Important part, glycolysis, first step of aerobic respiration, and then there's a preparatory step in the Krebs cycle, and then the electron transport chain. In glycolysis, let me see if I can blow this up. Glucose is the starting molecule right here. It is a 6-carbon molecule. ATP gets sent into the reaction in the preparatory step, and there's 2 of them, right here and right there. And then in the energy conserving stage, ATP gets generated. There's 2 here and 2 here. And glycolysis is 1, 2, 3, 4, 5, 6, 7, 8, 9, a 10-step process. This gets made, but then it's converted into that, and then it proceeds down. And glycolysis is a 6-carbon molecule. We end up with 2 pyruvic acids, which are 3-carbon molecules. Any question about any of that? For glycolysis, you do need to know the summary. 2 ATP enter the reaction. Of course, glucose enters the reaction. And 4 ATP come out. 2 pyruvic acids come out. And 2 NADH is come out. So the net gain of glycolysis is we get a net gain of 2 ATP, 2 pyruvic acids, and you can call that pyruvates. These are chemically similar molecules, and you need a chemist to note the difference. This has the hydrogen attached, this has the hydrogen released, and the cells treat these 2 molecules the same, so we will treat them the same even though they are chemically slightly different molecules. And then 2 NADHs are made by glycolysis. Okay, any question about any of that? So you need to know glucose comes in, you get a net gain of 2 ATP, 2 pyruvic acids are made, and 2 NADHs are made. Any question about any of that? All right, let's take a step back and take a look at the big picture. And that is, we're talking about aerobic respiration, but you do need to understand there's also anaerobic respiration. Respiration is the oxidation of molecules to release electrons, which are then given to the electron transport chain, and we'll talk more about that later. In the electron transport chain, that's where the majority of ATP is made in respiration. Now, aerobic respiration uses oxygen as the final electron acceptor. So the electrons that flow from glucose flow to oxygen in aerobic respiration. Anaerobic respiration is similar, except the final electron acceptor is another molecule other than oxygen. Any question about that? Well, what molecule it is depends on what form of anaerobic respiration we're talking about. There's many different forms. The final electron acceptor could be nitrite, nitrate, It could be a sulfate. It could be an iron compound or an iron, what do you call that? Iron element. The important part is in an anaerobic respiration, the electrons flow to another molecule that is not oxygen. And then in respiration, ATP is generated by the electron transport chain, which is shown here. Any question about any of that? All right. So we talked about glycolysis, where we convert glucose to pyruvic acid. Well, pyruvic acid cannot directly enter the Krebs cycle. So the pyruvic acid enters the preparatory step to prepare it for entry into the Krebs cycle. The preparatory step is just one step. Pyruvic acid is converted into acetyl coenzyme A. Pyruvic acid is a 3-carbon molecule. Acetyl coenzyme A is a 2-carbon molecule. So one of the carbons from pyruvic acid is burned and given off as CO2. In the preparatory step, we do make one NADH for each pyruvic acid. Now note, with one molecule of glucose, you generate 2 pyruvic acids. So for one molecule of glucose, the preparatory step runs 2 times. Is there any question about that? Okay, now the acetyl coenzyme A can enter the Krebs cycle. But you should know that the Krebs cycle has some different names. I, and your textbook author, tend to call it the Krebs cycle, after the doctor who discovered it. But it can be called the tricarboxylic acid cycle, or TCA for short, because the Krebs cycle involves a bunch of tricarboxylic acids. It can also be called the citric acid cycle, which has the same acronym TCA, because the first, the first tricarboxylic acid that's generated is citric acid in the Krebs cycle. Okay? Any question about that? Like I said, I'll be calling it the Krebs cycle. Your textbook author does. But there are different names for the Krebs cycle, and you really should know them. So this is showing you the preparatory step, which we've already discussed. Pyruvic acid is converted into acetyl coenzyme A, making an ADH. Acetyl coenzyme A then combines with, let's see if I can blow this up, combines with oxaloacetic acid to form citric acid. And then citric acid moves on to this molecule, which moves on to this molecule, which moves on to this molecule, which moves on to this molecule, and ATP gets generated right here. This molecule moves to that molecule, which moves to that molecule, which moves back to oxaloacetic acid, so we end up where we started. And that's why it's called a cycle. It just continues in a circle, ending up where we start, oxaloacetic acid. So as long as acetyl coenzyme A joins the Krebs cycle, it'll keep running. Any question about any of that? Now, as I stated, you do need to know the summary of the Krebs cycle as well as the preparatory step, which I've already talked about. You do generate in the preparatory step in the Krebs cycle 1 ATP, and I pointed that out, it's right here. You do generate 4 NADHs. There's 1 in the preparatory step, 1 right here, 1 right here, and then 1 right there for 4. You generate 1 FADH2 molecule, that's right here. And then for each of the carbons in pyruvic acid, each of the carbons is burnt into CO2. So we generate 3 CO2 from the preparatory step in the Krebs cycle. 1 happens here, 1 happens here, and the third happens here. Any question about that? All right, the NADH and the FADH2 carry the electrons from pyruvic acid and ultimately the electrons from glucose to the electron transport chain. And that's what NADH does in glycolysis too. It carries the electrons from glycolysis to the electron transport chain. All right, any questions about that? Here's a question for you. How many ATP are generated by the preparatory step and the Krebs cycle per molecule of glucose?
Three is what I want to say, but I feel like something else.
I didn't hear that. I'm sorry. Let me turn the volume up. For a molecule of glucose, we generate 2 ATP because the preparatory step in the Krebs cycle will run 2 times for each molecule of glucose, because glucose will be converted to pyruvic acid in glycolysis, and we generate 2 pyruvic acids. Okay? So for these numbers here, you need to multiply by 2 when we're talking about glucose. Is that clear to everybody? All right, I hope that's clear. This is showing you that NADH and FADH from the Krebs cycle and NADH carry the electrons from glucose to the electron transport chain. And in the electron transport chain, ATP production is kicked up into high gear. The electrons in aerobic respiration eventually flow to oxygen as the final electron acceptor. And then hydrogen ions, the electrons, and the oxygen are converted into water. So the electron transport chain, or ETC, is a series of carrier molecules that are in turn reduced and oxidized as electrons from glucose are passed down the chain. Energy is released as the electrons are transferred from high energy to lower energy compounds. The energy released can be used by chemoosmosis to produce ATP. Any question about that? Let's see if I can get that going. The electron transport chain has a couple of names. I'll always call it the electron transport chain or the ETC. Your textbook tends to call it that also. But it could be called the electron transfer chain, the electron transport chain. Generally, students don't get confused about that.
The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. Electrons captured from donor molecules are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain: NADH dehydrogenase, cytochrome BC1, cytochrome oxidase, and the complex that makes ATP, ATP synthase. In addition to these complexes, two mobile carriers are also involved: ubiquinone and cytochrome C. Other key components in this process are NADH and the electrons from it, hydrogen ions, oxygen, water, and ADP and PI.
There. Sorry, I had a hard time getting that to stop. I do want to tell you that most of the molecules you do not need to know. They named all of the
Two
molecules.
mobile carriers are also.
The only one you really kind of need to know is the ATP synthase molecule. The others you don't need to know. Let's start that again because I didn't show it.
Ubiquinone and cytochrome C. Other key components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, and ADP and PI, which combine to form ATP. At the start of the electron transport chain, two electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of one hydrogen ion for each electron. Next, the two electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. One hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradients animation. This animation illustrates 2 full cycles of electron donation. In biological systems, however, many electron transport cycles occur simultaneously, helping to ensure that the proton gradient is always maintained.
Okay, any questions? So let's let's look at that in a little more detail. In aerobic respiration, the electron transport chain is a chain of proteins in a membrane. In eukaryotes, the membrane, or where this occurs, is along the intermitochondrial membrane. So this is the inner mitochondrial membrane for eukaryotes. In prokaryotes, this membrane is the cell membrane, and it's called an electron transport chain because there's a chain of proteins in a membrane. Any question about that? It's called electron transport because the electrons are donated to the system. Let me blow that up a little. By NADH or FADH2, and then the electrons transfer or transport from one protein to the next protein, to the next protein, to the next protein, to the next protein, to the last protein. Okay? And then the electrons at the end, right here, combine with hydrogen ions and oxygen to form water. This is why you have to breathe. The oxygen, when you breathe, is converted in aerobic respiration into water. None of that oxygen is converted into CO2. Any question about any of that? That's why you have to breathe. And that's what happens to the oxygen. What happens to the water is we will breathe out a lot of it, but the organisms can conserve some of that and then use it for their water needs. And I've already talked that the NADH and the FADH2 come from the electrons in glucose, which flow to the electron transport chain from glycolysis, the preparatory step, and the Krebs cycle. Any question about any of that part? Now note that NADH is more efficient than FADH2, and that is NADH donates its electrons to the first protein in the chain of proteins. FADH2 does not donate to the first protein. It donates to a later protein. So NADH2 is more efficient at creating ATP than FADH2. And it's more efficient because some of the proteins in the membrane of the electron transport chain, when the electrons are moving through the protein, it gives the protein the energy it needs to pump hydrogen ions from this side of the membrane to that side of the membrane. And that's why NADH is more efficient. The first protein in the chain of proteins does pump hydrogen ions from this side of the membrane to that side of the membrane. So NADH pumps more hydrogen ions than FADH2. This protein and this protein also pump hydrogen ions from this side of the membrane to that side of the membrane when the electrons move through the protein. And the reason why they're doing that is, like I said, when the electrons are moving through the protein, it gives the protein the energy it needs to pump hydrogen ions from this side of the membrane to that side of the membrane. Any questions about any of that? That creates a two-fold hydrogen gradient. There will be more hydrogen on this side of the membrane than on this side of the membrane. There will also be more positive charges on this side of the membrane than this side of the membrane. That creates a twofold gradient of potential energy on this side of the membrane compared to that side of the membrane. These hydrogen ions will want to move from this side of the membrane to that side of the membrane. It's the law of simple diffusion. Hydrogen on this side, being higher in concentration, is going to want to move to this side. And then the positive charge over here is going to be attracted to, it's actually a negative charge on this side. That's not entirely obvious, but if you pump hydrogen ions from this side to that side, this side will have a positive charge, this side will have a negative charge. The only place the hydrogen ions can move from this side of the membrane to that side of the membrane is through this channel protein, which is an ATP synthase molecule or transport protein. And as the hydrogen ions move from this side of the membrane to this side, they give the energy from the potential energy difference. They give this protein the energy it needs to create ATP. And so this transport protein or channel protein is called ATP synthase protein. Okay, any question about any of that? And this is how, in aerobic respiration, the electron transport chain creates ATP by pumping hydrogen ions from this side of the membrane to that side, and then using that hydrogen ion twofold gradient to make ATP. And this is called chemiosmosis, but that's not a term that I will use further. Okay? All right, any question about any of this? So in aerobic respiration, the electron transport chain kicks ATP production into high gear. In eukaryotes, you generate 32 to 34 ATP for the electron transport chain. And it really depends on what cell it is. Most cells in your body produce 32 ATP per molecule of glucose in the electron transport chain. But there are 3 cells in your body which are more efficient, and they produce 34 ATP per molecule of glucose. And these 3 cells are cells in the heart, the liver, and the kidney. Why do you think your heart, your liver, and your kidney are more efficient at producing ATP than, say, a cell in your nose or a cell in your skin? Or
Maybe because, oh, sorry about that. So I would say because it's more, the heart, liver, and kidneys work a lot more.
Yeah, they're always working, assuming you're alive, and so they're more efficient. The cells on your skin don't work all the time, but the cells in your heart are constantly beating. The cells in the kidney are constantly purifying the blood, removing the nitrogenous waste. And the same with your liver. It's constantly cleaning the blood. It's constantly working, releasing glucose into the blood or taking glucose out of the blood if glucose is too high. Prokaryotes are a little more efficient, and they generate 34 ATP for the electron transport chain. Okay? And remember, in aerobic respiration, The final electron acceptor is oxygen, which combines with water to form, combines with hydrogen to form water. All right, any question about any of that? So where does the CO2 come from that you breathe out? It's not the oxygen you breathe in, because it's converted into water.
From the glucose?
Yes, we burn glucose in aerobic respiration, and each of the carbons in glucose is converted into CO2, or burned into CO2. So that's why we breathe out CO2, and then we breathe in oxygen so that we can convert that oxygen into water in the electron transport chain. All right, let's talk about this in a little more detail, and that is the NADH and the FADH2 carry the electrons from glucose to the molecules of the electron transport chain, and the electrons move from one protein in the membrane to another, releasing their energy slowly. And some of them, the energy is used to pump hydrogen ions across the membrane, and then the electrons flow to oxygen to become water, combining with the hydrogen ions. Let me reiterate that the electron transport chain happens in the inner membrane of the mitochondria for eukaryotic cells. So this right here is the inner mitochondrial membrane. For prokaryotic cells, this right here is the cell membrane. Let me blow this up just a little. Now, in eukaryotes, we're safe because when we pump hydrogen ions from this side of the membrane to that side of the membrane, that means we're pumping the hydrogen ions into the intermembrane space, and we have the outer membrane of the mitochondria to protect the cell from these hydrogen ions. Everybody see that? For prokaryotes, the cell membrane is where this electron transport chain is. And where are the hydrogen ions pumped in a prokaryote? Come on, it's in this picture here. The hydrogen ions are pumped from this side of the membrane to that side. Where are the hydrogen ions pumped to?
The cytoplasm?
No, they're pumped from the cytoplasm across the cell membrane to this side of the membrane. So the hydrogen ions are actually pumped into the cell wall. Okay? And then they will flow back across the cell membrane right here to make ATP. So those hydrogen ions are actually in the cell wall of a prokaryotic cell. Good guess, though. We've talked about that. I've talked about that. I've talked about that. And I mentioned the electron transport chain. The part of it making ATP is called chemiosmosis. So we already talked about that. We should say that the electron transport chain can also be called oxidative phosphorylation because phosphorylation is when you attach a phosphate group to a molecule and phosphate gets attached to ADP to make ATP. I will not use this term. I will always use the electron transport chain. But this is another term that could be used to describe the electron transport chain. So in aerobic respiration, the making of ATP from glucose, glucose is completely oxidized into CO2 via aerobic respiration, and we generate 36 to 38 ATP molecules per molecule of glucose in eukaryotes. Most cells generate 36 ATPs, but remember the heart, the liver, and the kidney generate 38 ATP molecules per molecule of glucose. Prokaryotes are a little more efficient. They always generate 38 ATP molecules per molecule of glucose. You should know the overall summary equation for aerobic respiration. Glucose, which is C6H12O6, plus 6 oxygen molecules and 38 ADP molecules, 38 inorganic phosphates, combine to make 6 CO2 molecules, 6 water molecules, and 38 ATP molecules. That's the overall summary reaction for aerobic respiration. And we're talking about in general for prokaryotes, and I suppose you could say the heart, the liver, and the kidney. For most other cells of eukaryotes, you only generate 36 ATP. Okay? Any question about any of that? And there's the summary of aerobic respiration in prokaryotes, starting with glycolysis, the preparatory step in the Krebs cycle, and then the NADHs and FADH2s carry the electrons to the electron transport chain, where the majority of ATP is produced in aerobic respiration. Any question about any of that? Any questions about aerobic respiration? If not, let's turn to anaerobic respiration. Anaerobic respiration is similar to aerobic respiration, with the exception that the final electron acceptor in ETC, the electron transport chain, is not oxygen. It's some other molecule other than oxygen. It can be an inorganic molecule like nitrate or sulfate. Anaerobic respiration usually has glycolysis and the Krebs cycle, so they are still occurring. The protein chain of the electron transport chain is present, but it's shorter in anaerobic respiration than in aerobic respiration. And because it's shorter, there is less ATP production in anaerobic respiration compared to aerobic respiration. Now, it depends on which form of anaerobic respiration we're talking about. The ATP yield can be between 2 ATP molecules per molecule of glucose, up to 36 molecules of ATP produced per molecule of glucose. And like I said, it depends on what form of anaerobic respiration we're talking about. Fermentation only generates 2, and I used to know 36. Let me see if I can remember that. I don't remember if that's a sulfate, anaerobic respiration, or a nitrate. I don't remember. But you don't need to know that because there's different forms of anaerobic respiration, and we're not going to talk about them. And that's why the ATP yield can vary so much. The important thing to know is that anaerobic respiration does not generate as much ATP as aerobic respiration in prokaryotic cells. Therefore, anaerobes tend to grow slower than aerobes because they produce less ATP. Any question about any of that? So here we're looking at respiration. In general, it's both aerobic respiration and anaerobic respiration. Generally, there's both glycolysis, the preparatory step in the Krebs cycle, and the electron transport chain. However, this electron transport chain is different for aerobic respiration and anaerobic respiration. The main difference is with aerobic respiration, oxygen is the final electron acceptor. With anaerobic respiration, it's some other molecule other than oxygen. And then the other thing we need to talk about is fermentation, which is another way that cells can use glucose to make ATP. Fermentation does produce ATP, but not much. Fermentation releases energy from the oxidation of organic molecules like glucose via glycolysis. Glycolysis proceeds as we previously discussed, and it produces 2 pyruvic acids, a net gain of 2 ATP, NADH and H+, from the oxidation of glucose. Any question about that? So fermentation always uses glucose to make ATP, and only 2 ATP are a net gain of glycolysis from glucose. We do generate some NADH, and then we attach 1 or 2 fermentation steps after glycolysis to ferment the pyruvic acid into the end product of the fermentation. And there are different fermentation reactions. We will talk about 2 of them and briefly discuss the others, but it's something like 5 or 6 fermentation reactions where you convert the pyruvic acid into the end product of the fermentation reaction. For lactic acid fermentation, pyruvic acid is converted into lactic acid. Fermentation uses the NADH created by glycolysis and then converts it to NAD+. Why do you think the cell, when it's undergoing fermentation, doesn't store the NADH up and then just wait until the NADH could be used for aerobic respiration? why does it use the NADH up in fermentation?
It need the hydrogen.
Yeah, you need the hydrogen for the fermentation, and then to continue the fermentation, you need to generate NAD+, because without any NAD+, you cannot engage in glycolysis. And the cell, like your muscle cells, if they are working and using fermentation to make ATP, because the oxygen is limiting in the muscle because the muscle is working so hard, the cell does want to continue making ATP from fermentation, and it has to regenerate the NAD+ in order for fermentation to go. Everybody see that? Well, hopefully everybody sees that because nobody's commenting. Okay? So for our muscles to continue to make ATP when they're really working hard, we do need to use up that NADH to generate NAD+ so that fermentation can continue. Because without any NAD+, we can't go further in glycolysis, and then we don't make any ATP, and if the muscle can't make any ATP, it will stop working. And then what will happen to the athlete? They'll just collapse. And I'm sure you've seen it. Almost at every Olympics, there's an athlete that works so hard trying to cross the finish line that they run out of, they probably run out of glucose, okay? And they stop making ATP from the fermentation in their muscles, and their muscles just collapse, and they collapse. In the wintertime, their coach will run out and throw a blanket over them. I'm not sure what they do in the summertime. They probably run out with a squirt bottle and then spray them down. And they will be fine because their heart and the liver, not the heart and the liver, the heart and the lungs are trying to get oxygen to the muscle, but it can't get enough oxygen to the muscles, and that's why the muscles are engaging in fermentation. But as the heart and the lungs bring blood to the muscles, they'll bring glucose to the muscle, and then the glucose can be used to make ATP by fermentation, but also the heart and the lungs will bring oxygen to the muscles, and then the muscle can use aerobic respiration to make ATP. The athlete will then get up and then cross the finish line. And like I said, that usually happens every Olympic athlete, or every Olympics, to one athlete where that happens. Any question about any of that? All right, so this is showing you lactic acid fermentation, which our muscle cells can do. It's also how we make lactic acid in yogurt, sauerkraut, in most cheeses like cheddar cheese, every cheese you can think of except for Swiss cheese. Yes. And a bunch of other foods like chocolate and other fermented food engages in lactic acid fermentation, where glucose is converted to pyruvic acid by glycolysis, making ATP, and then pyruvic acid is converted into lactic acid in one step. Okay? And you can feel the burn in your muscle when the lactic acid builds up in your muscle. And the muscles do fermentation only when they're working so hard that oxygen becomes limiting. And so aerobic respiration cannot proceed. So the muscle engages in fermentation to make ATP so the muscle can continue working. Any question about that? And like I said, this is also how we make most cheeses, yogurt, sauerkraut, and most other fermented foods. Alcoholic fermentation, also called ethanolic fermentation, is how we make beer and wine, as well as sourdough bread. It engages in glycolysis to make ATP, and then the pyruvic acid is fermented in 2 steps to ethanol. Okay? You'll note that ethanol is a smaller molecule than pyruvic acid. Pyruvic acid has 3 carbons. Ethanol has 2 carbons. So 1 carbon comes off. When you drink your beer, this carbon gives the beer the fizz. We make wine in an open barrel, so the CO2 gets vented to the air. But if you were to bottle the wine, it would be fizzy, and we call that champagne, fizzy wine, because of the CO2. Any question about that? The CO2 in sourdough is given off and causes the bread to rise. And that's alcoholic fermentation. There are different fermentation reactions, and this chart is showing you them. We've already talked about lactic acid fermentation and ethanolic fermentation. You'll notice that Propionibacterium produces propionic acid, which is how Swiss cheese is made. It's a different fermentation reaction than all the other cheeses which use lactic acid fermentation. There are different fermentation reactions which can be used by industry to make different products like acetone and isopropyl alcohol, lactic acid, succinic acid, acetic acid. And then Enterobacter, we use that to make, what do you call that? When you have wine, it can go bad and form, what is that product called? It's an acid, nobody knows. I don't remember. Formic acid? That doesn't sound right. I'm losing it. Let's move on to the next slide, and hopefully I've got it on here. There's Swiss cheese, propionic acid fermentation. I'm not seeing what I'm looking for. Acetic acid, but we don't call it that. What do we call that?
Vinegar.
Vinegar, thank you. I couldn't remember. Yeah, wine can go bad and form vinegar. There it is, acetobacter and vinegar. It was right there, I just couldn't find it. That's from acetobacter growing in the ethanol. Okay, making vinegar. Thank you for helping me there. Anyway, it's showing you the different industrial uses. You don't need to know this slide. You should know about ethanolic fermentation and lactic acid fermentation. These are the main ways that we make beer, wine, bread, at least leavened bread, and then most of the fermented foods that we eat, like cheese, most cheeses, yogurt, rye bread, sauerkraut, and sausage as well. The exception is Swiss cheese, and I'm not going to ask you to know about that one. All right, any question about fermentation? You should know that, oops, a little too far. A summary reaction of aerobic respiration, anaerobic respiration, and fermentation. Aerobic respiration is the most efficient at producing ATP. You can get up to 38 molecules of ATP in a prokaryotic cell per molecule of glucose. Oxygen is the final electron acceptor. In anaerobic respiration, the final electron acceptor is some molecule other than oxygen. You get fewer than 38 molecules of ATP, but more than 2 molecules of ATP. Fermentation, we only get 2 molecules of ATP per molecule of glucose. And the final electron acceptor is actually an organic molecule that's used by the fermentation reaction. Okay, so pyruvate will take the electrons and then be fermented to some other molecule in fermentation. All right, any question about this slide? Now, we've talked about carbohydrate catabolism and how our cells can use it in aerobic respiration or fermentation to generate ATP. You do have to realize that your cells can use more than carbohydrates. They can also use lipids and protein catabolism to make ATP. So everything you eat as food can be used to make energy in the form of ATP. And this happens with microbes as well. There is a problem in that large molecules like protein or even a lipid cannot cross the cell membrane. And microbes, as well as your intestinal cell, produces lipases and proteinases to extracellularly digest those large molecules and break down the large ones, the proteins and the lipids, down into monomers, and then bring the monomers into the cell and then utilize them by catabolism to make ATP. Okay, so the protein will be digested by extracellular proteases in both microbes and in your cells to make amino acids, and then the amino acids can be transported across the cell membrane, and then the cell can digest them. With amino acids, the amino group has to be removed, and then the remaining part of the amino acid is converted into something to enter the Krebs cycle, and we'll show that in the next slide. The lipids are broken down into organic acids, and then they're broken into 2 carbon chunks, which then directly enter the Krebs cycle. Let me see if I can blow this up, showing you the lipid and protein catabolism and the interrelationship with carbohydrate metabolism. So proteins will be broken down into amino acids, and depending on which amino acid they are, the amine group will be pulled off, and for us that'll be a nitrogenous waste which will be urinated out, and whichever amino acid it is, it can enter the glycolysis low down in the reaction of glycolysis, or it can enter and be converted into acetyl coenzyme A, or the amino acid can enter into the Krebs cycle, depending on what amino acid we're talking about. Lipids, the glycerol will be broken off and it will be entering into glycolysis. The fatty acids will be broken down into 2 carbon chunks, and they'll be converted into acetyl coenzyme A and directly enter the Krebs cycle. Okay? Now for couch potatoes, we will actually break these down, and these down too, and most of the time we'll convert those molecules into glucose. And the same thing with carbohydrates, like you eat starch, it'll be converted into glucose, and then glucose will enter aerobic respiration in the normal way. Okay? So couch potatoes will do that. But if you're a marathon runner, you won't do that. And the lipids will be broken down into glycerol and fatty acids and will directly enter the Krebs cycle or the glycolysis. Okay, any question about that? The point is, with metabolic pathways, for using energy, the oxidation of molecules, including glucose in aerobic respiration, is the most efficient, and then it uses glucose to make ATP. And then most cellular ATP is used later by the cell for its energy needs. An example would be the biosynthesis of large molecules like polysaccharides, lipids, proteins, and nucleic acids, the 4 molecules of life. And this we call breaking down our food molecules, especially glucose, but anything we eat, and then sending it through aerobic respiration. We call that anabolism for generating ATP. Let me mention that you don't have to use catabolic did I say that right? I did not say that right. The breaking down of glucose and our other food molecules, that is catabolism. And that energy we make as ATP is usually used in anabolism, like the biosynthesis of larger molecules. But the ATP could be used to transport a molecule around in the cell, for the muscle to move, for the cilia to move the cell, or the, what do you call it, the flagella to move the cell. And we've talked about anabolic and catabolic pathways as separate, but they are joined by common intermediates. And an amphibolic pathway is where the metabolic pathway can function in both directions. They bridge the pathways involved in carbohydrate, lipid, and protein and nucleic acid metabolism. This slide is trying to show you, let me see if I can blow this up. The amphibolic pathways involved in the metabolism of molecules in aerobic respiration. Wherever you have two lines, you can have an amphibolic pathway, meaning it can go in the opposite direction. The important part is that carbohydrates can be converted into glucose and then enter aerobic respiration, but you can see amino acids can be converted into glucose, and lipids can be converted into fatty acids, and actually they enter acetyl coenzyme A directly, converted into acetyl coenzyme A. But if we have lots of carbohydrates that we're consuming, we'll convert that into fatty acids and then store that down as a lipid. And my point is that even animal cells, we have an awful lot of amphibolic pathways where the reaction can go both ways. Any question about any of that? Green plants are much better at amphibolic pathways than we are. All right. And that is that they can make all of the molecules they need, generally from carbon dioxide and water. Animal cells are nowhere near as good at doing that, but generally animal cells can convert proteins to carbohydrates or lipids to carbohydrates, and they can also convert protein or carbohydrates to lipids, and vice versa. Any question about any of that? All right, we've talked about catabolism for the generating of ATP, the degradation of, where do I have that? The degradation of a molecule like glucose to make ATP. But we don't have to use catabolism to make ATP. In photosynthesis, we can make ATP using not a catabolic pathway, but an anabolic pathway. Photosynthesis is the conversion of light energy into chemical energy, including ATP. And so we say it's the conversion of light energy into chemical energy. The chemical energy may be used to convert atmospheric carbon dioxide to glucose. And so usually the ATP in photosynthesis will be used to make glucose, but it doesn't have to be, and hopefully I'll have enough time to talk about that. Remember, photosynthesis happens in more than just green plants. It happens in all algae, including green algae, and it happens in cyanobacteria, the photosynthetic bacteria, which cyanobacteria makes oxygen. Photosynthesis can also happen in bacteria that do not make oxygen, but we're not going to talk about that now. Anyways, photosynthesis is a way of synthesizing organic compounds, like glucose, which animals and microbes need to feed on. And they get these molecules from other molecules, such as green plants. Photosynthesis occurs only in photosynthetic organisms. These organisms contain a light-trapping pigment. You actually know one of them, and it's chlorophyll. The light in photosynthesis releases the electrons from chlorophyll. Those electrons flow and then can be used to make ATP or NADPH, which are often used up by the cell to make glucose from CO2 and water. We say the making of glucose from CO2 and water is carbon fixation. There are two different photosynthetic reactions. You've probably only heard of non-cyclic photosynthesis. In non-cyclic photosynthesis, we combine 6 carbon dioxide, 12 waters in the presence of light to make glucose, C6H12O6, 6 oxygen molecules, and 6 water molecules. You need to know this chemical reaction for non-cyclic photosynthesis, and you can subtract 6 water molecules here and 6 water molecules here, but I prefer showing all 12 because we look at where the 12 oxygen molecules come from in photosynthesis. All of the oxygen, the 12 atoms, come from the water. Okay? But there is another photosynthetic reaction that we call cyclic photosynthesis. And in cyclic photosynthesis, we do not make glucose or oxygen. Instead, in cyclic photosynthesis, you make ATP from ADP and phosphate. All photosynthetic cells can engage in both cyclic photosynthesis, which they do if they need ATP, or they can engage in non-cyclic photosynthesis if they want to make glucose. Is that clear to everybody? Okay, we're out of time. I have a couple more slides to show, and I think we're going to discuss them next time. Are there any questions? If not, I'll see you at 6:30 for the lab. If you have questions, if you don't have questions, you do not need to show up for the lab. I will log off at 6:45 unless there's a whole bunch of questions, in which case I'll log off when the questions get answered. All right, I'll see you later.
Thank you.
Bye.